My name is Michael Vega, and I'm a PhD candidate in the Environmental Engineering Department. Today I'll be talking to you about my research looking at sulfamethoxazole biotransformation, sulfamethoxazole being a pharmaceutical that's persistent in wastewater, and how it's linked to methane oxidation in an engineered wetland biomat that treats a wastewater-impacted river. So the last few decades, there's been a lot of research trying to understand the extent to which pharmaceuticals or trace organic compounds are accumulating in wastewater treatment plants. So the basic idea is that as humans, we ingest a lot of pharmaceuticals or pills, and our body doesn't completely break them down. So we excrete them as waste, and they end up in wastewater treatment plants. And a lot of research has found that conventional wastewater treatment is not exceptionally good at removing these compounds, and so they end up getting released into the environment. This is extremely bad. Unfortunately, we don't really understand, at least not thoroughly, a lot of the mechanisms or processes or pathways by which these compounds are degraded in wastewater treatment, and this is a big issue, so we need to understand this better. More specifically, the problem that my research is trying to address is that we know very little about how microbes or microbiology contributes to the degradation of these pharmaceuticals in wastewater, but even less so in the environment. But one thing that the research has taught us recently is the role of this enzyme here. This is ammonia monooxygenase. It's an enzyme that bacteria produce to catalyze this chemical reaction and grow. Basically, the oxidation of ammonia to hydroxylamine using oxygen as an electron acceptor. And this is a really important enzyme in conventional wastewater treatment because it catalyzes the first step of wastewater treatment, which is nitrification, this chemical reaction here. Recent studies in the past 10 to 15 years have shown that the non-specificity of this enzyme allows compounds that are similar to ammonia to sort of slide into the active site and reap the benefits of the enzyme. And this is called cometabolism. So basically what that means is that the organisms are still growing with ammonia as their primary substrate, but other compounds are able to slide in and reap enzymatic benefits. The system that I work on is quite different in a number of ways. First and foremost, it's a constructed wetland. So it's a passive biological treatment system that's designed to emulate natural processes to remove contaminants from waste streams. And so in addition to that, it's a constructed wetland, right? And so as you see in this image here, it probably doesn't look like a wetland that you're used to seeing because there's no plants. So a little bit more about the system. It's a shallow open water wetland, and so it's deliberately designed to prevent plant growth, and so there's a geofabric along the bottom that prevents root development and plants don't grow. When you combine that with a foot-deep water column and a nutrient-rich influent, you get this microbial community or biofilm or biomat that develops along the bottom that can be as thick as 15 centimeters, so extremely thick. And you can see from these images that it's redox stratified, where it's photosynthetic and green and diffuse at the top, and then cake-like and anoxic at the bottom. The system is also very different from conventional wastewater treatment plants in that it receives river water that is impaired by already treated wastewater, and so it's nitrified wastewater effluent. What that basically means is that all the ammonia in wastewater has already been converted through nitrification, the process I discussed earlier, to produce nitrate. And so there's not really any ammonia going in, and we wouldn't expect ammonia monooxygenase activity for that reason to be a predominating biological process. However, there's a highly analogous enzyme called the particulate methane monooxygenase, one of two methane monooxygenase enzymes that bacteria called methanotrophs use to perform this chemical reaction, where methane is oxidized to methanol using oxygen as an electron acceptor. And based on similarities between this and ammonia monooxygenase ammonia, methanotrophs are also capable of oxidizing ammonia to hydroxylamine, and nitrifiers, bacteria that oxidize ammonia, are also capable of oxidizing methane. And this has been known for several decades. Similarly, studies have found late 80s to early 90s that other compounds like halogenated hydrocarbons, in this case TCE, a potent carcinogen, can also be co-metabolically biotransformed by the particulate methane monooxygenase. And this has been studied quite extensively since then. But what about pharmaceuticals? So this hasn't been looked at in the context of pharmaceutical biotransformation for two reasons. One, because pharmaceutical biotransformation pathways are only recently being studied, the last 10, 20 years. And two, because methane monooxygenase activity is not a predominating enzymatic pathway that is found or observed in conventional wastewater treatment. So the broad hypothesis that my research is trying to address is that biomat PMOMO activity, or when bacteria that oxidize methane produce these enzymes contribute to the biotransformation of pharmaceutical compounds. So now I want to take a step back and go through a conceptual framework for biomethane cycling and discuss some high-level microbial community data that suggests that methanotrophs or methane oxidizing bacteria are abundant, potentially active in the superficial oxic layers. So here on the left, we have this conceptual image, conceptual diagram, highlighting where methane production and methane consumption might be observed in the biomass. So we would expect to see the most methanogenesis under anoxic layers, the black layer down here, performed by methanogenic archaea. And then as that methane bubbles up, when it reaches the oxic layer, we would expect to see methanotrophic bacteria, or methane oxidizing bacteria, to use methane monooxygenase to catalyze that reaction. And we can use DNA and RNA sequencing to look at the relative abundance of lineages or microbes that are capable of methane oxidation as a function of depth and whether or not they're potentially active. So on this graph, we have cDNA relative abundance on the y-axis and DNA relative abundance on the x-axis. And basically anything that plots above this one-to-one line can be inferred to be potentially active because it's more represented or of more abundance in the RNA-based cDNA microbial community. And so when you look at the data here where the colors denote depth, the lighter colors are higher depth, you can see that methanotrophs are generally more abundant, more potentially active in these superficial layers where we have oxygen, where we would expect there to be methanotrophic activity. And so getting back to the trace organics, here we have the 6 compounds that we chose to survey, broken into classes: antibiotics, beta blockers, anticonvulsants, and antivirals. For this talk, I'm only going to have time to discuss sulfamethoxazole data. So looking at pore water data from the field, we collected pore water within a mature open water wetland biomat using this adapted mini point sampling technique courtesy of the USGS. And as you can see in the graph on the left here, we have sulfamethoxazole as a function of depth, where the colors denote different sampling times. You can see that sulfamethoxazole is generally degraded in the superficial 2-ish centimeters, and that deeper than that, the only depth that we had for this field campaign was at 6 centimeters. You see that there was pretty much non-detectable sulfamethoxazole. So from this, we can broadly conclude that sulfamethoxazole is degraded in this superficial 2 to 3-ish centimeters of the biomat. And so when we couple this with the microbial data suggesting that methane oxidation is potentially occurring in the shallow biomat layers in parallel with sulfamethoxazole degradation, you have to ask the question: are these things related? Because this is very much associative field scale data. So we went back into the lab and addressed this hypothesis again to more functionally and mechanistically understand whether or not these processes are linked. So I conducted some microcosms in serum bottles with 2 grams of biomat and 160 mils of water, where I tried to induce a methane concentration gradient. So from left to right here, we have increasing methane concentration, and then from top to bottom, I amended with different inhibitors to inhibit methane monooxygenase activity. And so the 40,000 foot takeaway for this experimental design is I was trying to induce methane oxidation across a concentration gradient, and then use selective inhibitors to inhibit either both methane monooxygenases or just one methane monooxygenase to see how enzyme activity relates to sulfamethoxazole biotransformation. And so when we look at the experimental data for methane consumption, here we have a plot of cumulative methane consumption on the y-axis in time, on the x-axis where the different colors denote different aqueous methane concentrations, and the shapes denote the either inhibitors or no-MAT controls or uninhibited conditions. You'll notice these arrows designate times when methane was fully consumed and I had to refeed methane to the bottles. But the first big takeaway that you see from this graph is that we had a very clear methane oxidizing gradient as a function of the amendments that we had. And you also notice that the inhibited and no-MAT controls saw very limited methane consumption or methane oxidation. You also notice that the no-MAT controls were not significantly different from the inhibited bottles, which suggests that the inhibitors did a really substantial job in suppressing methane consumption. Lastly, you'll notice that between the 2 inhibitors, which have different effects on the methane monooxygenase enzymes, there wasn't a significant difference. And so from that, because allylthiourea only inhibits one methane monooxygenase, it's a particulate methane monooxygenase, we can infer that biomat methane oxidation is mediated by the particulate methane monooxygenase. And so when we look at this in comparison to sulfamethoxazole biotransformation profiles, here we have the same methane consumption graph on the left and sulfamethoxazole as a function of time on the right using the same legend. You can see that sulfamethoxazole is only biotransformed in the presence of methane oxidizing activity, and there's extremely limited, if any, biotransformation in the no-MAT controls and inhibited conditions. So we see this really clear, almost dependence of sulfamethoxazole biotransformation on methane oxidation. So more broadly, what we can conclude from this is that in open water wetlands, sulfamethoxazole biotransformation appears to be dependent on methane oxidation. And so we're still grappling with what this might mean and the broader ramifications for engineering applications or system evolution and function. But this is something that we can broadly conclude at this point. Zooming out even further, thinking about the applications of these findings to natural wetlands, provided it's a natural treatment system, and the fact that natural wetlands are a global methane reservoir, and knowing that methane is a potent greenhouse gas, about 30 to 40 times the global warming potential of carbon dioxide, and also recognizing that methanotrophs and methanotrophic activity, methane oxidizing activity, is ubiquitous in these systems. Does this mean that methane-rich ecosystems like natural wetlands are an unaccounted for sink for sulfamethoxazole? This is something that we're actively thinking about. And so with this, I'd like to acknowledge my funding sources, and thank you so much for your time in watching this talk, and thank the organizers for putting everything together. And if you have any questions, don't hesitate to email me or contact me on Twitter. Thanks so much.